Welcome to the MS&Me Radio Network. The broadcast shows are for informational and entertainment purposes only. They are not designed to provide listeners with specific personal, medical, or counseling advice. Individuals with a medical issue should always consult their healthcare provider. MS&Me is not responsible for content of individual shows. The views expressed by show hosts or their guests are their own. Enjoy the show.
I'm John Strum, and this is RealTalk MS. It's June 4th, and we have a lot to talk about. And after spending the month of May flying over 27,000 miles to attend MS conferences and meetings in Washington, D.C., Philadelphia, Copenhagen, and Seattle, I'm especially happy to be talking to you from the comfort of home. Last week, I was in Seattle among the more than 2,000 healthcare professionals attending the Consortium of Multiple Sclerosis Centers annual meeting. And while almost all of the MS conferences tend to focus on research, the CMSC annual meeting is the only conference that highlights the latest treatment strategies for MS healthcare professionals. So you'll find a mix of neurologists, nurses, social workers, mental health professionals, and rehabilitation specialists all in attendance, looking for ways to improve the quality of care for people affected by MS. Over the course of 3 days, there was a mix of lectures, expert panel presentations, and education programs that covered everything from the fundamentals of MS care to current trends in MS rehabilitation, sexuality and intimacy in MS, pediatric MS, the future of MS imaging technology, mental health issues in MS, and a lot more. My biggest challenge each day of the conference was trying to determine which sessions to attend. And I'll admit there were times that I found myself splitting sessions, seeing the first half of one presentation and then dashing out of one lecture hall and sliding into another in time to catch the second half of a different presentation. This conference also gave me the opportunity to get face-to-face with some of the people who I ordinarily connect with by email, and that was a real treat. If you haven't yet had the chance, I hope you check out the daily bonus episodes that I produced from the CMSC annual meeting. I'll include a link to each of them in today's show notes. And today, I'll be announcing the winner of our contest to be my special guest on the 100th episode of Real Talk MS. This is episode number 92, so our 100th episode is right around the corner, and I want to thank all of you who submitted your story. I'll be announcing the winner a little bit later in today's podcast, and our winner's going to be joining me on Real Talk MS episode 100. Since I spent all last week on the healthcare delivery side of MS, today's a good day for us to talk about MS research. My guest today is Dr. Jeffrey Huang. Dr. Huang is an associate professor in the Department of Biology at Georgetown University and an MS research scientist. We're going to talk with Dr. Huang about his potentially breakthrough research, and I'm going to ask him exactly how he became an MS research scientist. But before we get to my conversation with Dr. Jeffrey Huang, there are a few other things that you should know about. One of my favorite parts of most any MS conference is the poster session. Graduate and postgraduate researchers publish large posters that summarize a specific project that they've worked on. And during specific times of the day, a member of the research team will stand next to their poster, ready to answer any questions that people might have about their project. One of the posters that I came across last week at the Consortium of MS Centers annual meeting summarized a survey about the preferences, needs, and opportunity areas for MS caregivers in the United States. And if you know me, you already know why this poster attracted my attention. Reading the material in the poster, one of the first things that I noticed about this survey was that while almost 1,000 people responded to the survey, 896 of the respondents were people living with MS. Only 171 caregivers actually participated in the survey, And when you consider that the objective of this survey was to better understand the challenges experienced by caregivers and to identify effective strategies to support caregivers, I would have hoped that the survey would have actually engaged with a higher number of caregivers. Having said that, it was interesting to see that the people living with MS and the MS caregivers seemed to be in large agreement about the tasks and activities that were most useful to the person living with MS. When the research team analyzed the perspective and perception of the MS caregiver, the survey showed that only about half of the MS caregivers felt that they were a part of the MS patient's care team. And this shows a pretty strong disconnect between the caregiver and the care team. My experience as an MS caregiver has taught me that you sometimes have to remind some healthcare professionals, and sometimes you have to forcefully remind some healthcare professionals of your role as a member of that care team. You have a virtually unlimited amount of data to share about their patient, and your insights can't ever be replaced by diagnostic tests. From my perspective, MS caregivers can add to a patient's profile in unique ways that just can't be duplicated or substituted. If you wait to be invited onto your family member or loved one's care team, you may be waiting a very long time. Instead, I recommend that you claim your role as a member of that MS care team, adding value to the conversation every step of the way. Your insights can help fine-tune any treatment plan. And when we fast forward to the conclusions that were reached by the research team, well, the initial conclusion is that there's a lack of resources supporting MS caregivers. And once again, as someone with a significant amount of experience as an MS caregiver and as someone who's spoken with a lot of MS caregivers, I have a slightly different perspective. It's my experience that the challenge facing MS caregivers isn't a lack of resources. The challenge is that there aren't enough MS caregivers who are connected to or aware of the support resources that are available to them. Well, that's why I'm involved in a research project through iConquer-MS that will suggest a protocol for proactively connecting MS caregivers to the support resources that are designed to improve their quality of life as caregivers, and in doing so, improve the quality of life for the people living with MS for whom they're caring. So I apologize for the mild rant here, but it seems that too often the research studies that focus on caregivers all reach the same or similar conclusion, that caregivers need more support resources. And by now, there are plenty of support resources, just not enough caregivers with an awareness that they exist. And I'm hoping to see us take a small step in a new direction to raise that level of awareness. And if you'd like to see what one of these posters looks like and how it's all laid out, well, I'll include a link to the poster for this particular study in today's show notes. Over the past few years, the role of the gut and the bacteria in the gut, or the gut microbiota, has been shown to have some sort of connection to autoimmune diseases like MS. And an Israeli research team has demonstrated that social stress changes the gut microbiota of mice who are genetically susceptible to autoimmune disease. And that change in the gut bacteria triggers an expansion of self-reacting immune cells, or an autoimmune response. The authors of this same study also point out that some of this bacteria that gets triggered in mice when they're exposed to external stress has also been found in very high numbers in the gut of people with MS. As science has already focused on understanding the connection between the gut microbiome and multiple sclerosis, this study sheds some light on one interesting phenomenon. There's still a lot left to understand about our gut bacteria and its connection to our central nervous system. And if you'd like to get the details of this study, you'll find a link in today's show notes. The results of a phase 2 clinical trial published in 2014 demonstrated that simvastatin, an oral statin marketed under the name Zocor, reduced brain atrophy improved cognitive function, and even slowed MS disease progression when compared to a placebo. Today, statins are routinely prescribed to people at risk of heart disease. Statins lower the amount of LDL cholesterol, or so-called bad cholesterol, in the blood. But besides lowering the levels of fat molecules in the blood, statins also have anti-inflammatory and neuroprotective properties, and those characteristics make statins likely candidates as a treatment for MS. In fact, following the Phase 2 clinical trial, a large Phase 3 trial is now underway at multiple sites in the UK to determine how well treatment with simvastatin might slow disease progression in people living with secondary progressive MS. But the one loose thread that's been hanging around from that Phase 2 clinical trial was the question of whether simvastatin was effective in reducing brain atrophy and slowing MS progression as a function of statins' effects on blood cholesterol levels, or if there were other mechanisms at work here. So University College London researchers designed sophisticated mathematical models to analyze the data to better understand those positive outcomes in their Phase 2 clinical trial. And they found that simvastatin's positive effects on secondary progressive MS were independent of its effects on cholesterol. Their statistical analysis illustrated that treatment with simvastatin worked differently for people with MS than it did for people who needed to lower their cholesterol. Based upon what researchers learned during the Phase 2 clinical trial, the Phase 3 trial currently taking place may point the way toward a promising and effective treatment for people living with secondary progressive MS. I promise to keep you posted on this particular study. And if you'd like to review the statistical analysis of the Phase 2 results, I'll include a link in today's show notes. Biopharmaceutical company AbbVie has teamed up with MC10, a wearable sensor maker, to launch a new set of clinical trials using MC10's FDA-approved wearable device system, the BioStamp Endpoint System, to keep track of each patient's physiological data and outcomes during the trial. There's been a lot of interest in how wearable digital technologies might be leveraged to improve the design and development of clinical trials. The idea is that these technologies can provide a research team with a broad range of data while eliminating the patient's responsibility to report their outcomes. And it should be noted that these types of wearable digital devices aren't the same as your Fitbit. In order to qualify for use in a clinical setting, these devices require rigorous FDA approval. MC10 has also partnered with UCB for Parkinson's disease research. Better science can produce better outcomes. Improving the quality of data collected during a clinical trial improves the ultimate outcomes and conclusions that can be drawn from the trial, and you can expect to see more companies making these kinds of announcements in the very near future. If you'd like your own guided tour of MC10's BioStamp Endpoint System, which has been designed so that each patient can use it in their own home, please be sure to check out the video that you'll find in today's show notes. We focused a lot on research in today's podcast episode because I believe that good science is the engine that creates the future. Whether we see clinical trials ultimately succeed or fail, we hopefully learn something along the way. And that's the progress that fuels hope. Getting a diagnosis of a progressive neurodegenerative disease like MS isn't something that's easy to live with. That's why I try to give you at least a small heads up that good things are happening in laboratories around the world. One of those laboratories is at Georgetown University, where my guest, Dr. Jeffrey Huang, is at work on what one day might become a game changer in the treatment of multiple sclerosis. In a moment, we'll meet Dr. Jeffrey Huang and find out what he's been working on. And after my interview with Dr. Huang, we'll find out who is going to be my special guest on episode 100 of Real Talk MS. My guest today is Dr. Jeffrey Huang. Dr. Huang is a research scientist at Georgetown University, and he's a recipient of the Harry Weaver Neuroscience Scholar Award from the National MS Society. Dr. Huang received the Weaver Award to continue his research on what I think is a potentially groundbreaking treatment for MS. Welcome to the podcast, Dr. Huang.
Hi, thank you, John, for inviting me, and I'm very happy to be part of this podcast.
Well, first, let me congratulate you on receiving the Weaver Neuroscience Scholar Award. I know your research involves understanding the role of a particular molecule in myelin repair and its potential as a target for remyelination or restoring function for people living with MS. But before we get into the specifics of your research, I thought it might be interesting for our listeners to get an idea of how someone becomes a scientist doing MS research. So can you share with us a little bit about how you got here? How does someone wake up one day to find themselves in a lab doing the kind of work that you're doing?
All right. Well, that's a very good question, actually. I sort of got into this MS research in some ways sort of gradually. I started my PhD when I was a graduate student at Mount Sinai School of Medicine, where I worked with a scientist who was very interested in simply the biology of myelin and how myelin is generated and how does it wrap around the nerve fibers. And I was really struck by, sort of how beautiful the structure of myelin was around axons, because if you look at it under an electron microscope, you see this really nicely spiraling structure around the axon that is really, for me, I was just curious how it wraps. And so simply through its an appreciation of its structure and wanting to understand sort of the biology and the cell biology mechanisms of how myelin interacts with axons and wrap around it, was what got me really excited and got me started into this area of research. And then I was very fortunate to have, after my PhD, to move to the University of Cambridge, where I worked with a really well-known scientist in the MS and remyelination field. And I sort of learned a lot over there about the clinical relevance of understanding myelin, how myelin is destroyed in MS and how it potentially is an area that needs to be targeted in MS therapy. So that's really how it started. And in terms of how I got into MS research in Cambridge mainly, and it was, what was fascinating to me was not just how nice the structure was. What was fascinating to me is the fact that myelin actually could regenerate. And, this was something I didn't really appreciate when I was a graduate student. So when I went to Cambridge, I realized and learned that, the brain has these neuroprecursors that become activated following injury, and they're able to regenerate into myelinating cells. And then this has significant relevance in multiple sclerosis research, because as we know, myelin and the cells that make myelin, the oligodendrocytes, are destroyed through autoimmunity and inflammation. So the fact that they could regenerate tells us that we could potentially target the regenerative mechanism of these precursor cells to promote repair in multiple sclerosis. So from there on, I moved on and started my own lab at Georgetown University, and really started to focus and look into the mechanisms of how remyelination is achieved following experimental injury, so that we could potentially develop therapeutic and drugs for MS.
Tell us a little bit about the work that you're doing now, some of the specifics about the research project, because it sounds amazing to me.
Oh, well, thank you. I appreciate that. We're mainly interested in, how do oligodendrocytes regenerate following injury? So we try to understand the mechanism, right? So what are the key players, the molecules? What is the role of the environment, in the lesion that could promote or prevent the repair process, so that we could further understand and sort of relate this to multiple sclerosis and further understand how we could promote repair in MS. So in order to do this, our lab is focusing on animal models of multiple sclerosis, and we could experimentally demyelinate, perform demyelination in the mouse central nervous system, and then we could track the regenerative process over time. So because we could track the regenerative process, we are now looking at all the molecular players and as well as the biochemical players in the lesion environment that's involved in repairing myelin. So yeah, basically that's what we're doing. So there are a number of potential drug targets as well as important molecules that we have already identified. And so we're testing the effect of these molecules and drugs on remyelination in our animal model, with the hope that we could potentially identify novel drugs and to take it to the clinic.
Just to make sure I'm clear, have you been able to promote remyelination in the mouse model of MS?
Yes, there are. Yes, so even when I was a postdoc, we identified at the University of Cambridge, we identified a potential drug target that would, has, at least in mouse models, was able to promote remyelination. When I moved here, we started to look at other factors, particularly factors associated with inflammation and the role of inflammation in remyelination. The reason why this is important is because in multiple sclerosis, this is characterized by chronic inflammation, so sort of the activation of immune cells, not just in the blood, but in the brain as well. So these immune cells are activated, and if you can't control that inflammation, then we found that the regenerative response becomes impaired. So since starting my lab here, we've identified a number of molecules that are involved in inflammation modulation in the brain. And we found also that there are, so mice that are deficient in the expression of these molecules have aggressive inflammation. And if we could use, we've also found drugs that could potentially target inflammation to sort of modulate the inflammatory environment in order for remyelination to occur. So that's sort of the kind of projects that we're doing at the moment.
And to help my listeners kind of better see the potential outcome here, at least in the mouse model at this point, the mice who experienced remyelination, they basically had a high degree of recovery. Is that correct?
Yes, that's true. So there are a number of different models that are used in MS research. So we use different mouse models and to capture the different aspects and pathology of multiple sclerosis. And one of the models that we use is this focal lesion model, where we essentially could experimentally induce a lesion in the spinal cord or in the brain of a mouse. And then following this injury, they spontaneously repair over a course of about 30 days. And here, if we treated these mice with this drug, with drugs that we have identified, we're able to promote remyelination or accelerate the repair process. The other model that is commonly used, and we use as well, is called experimental autoimmune encephalomyelitis. Essentially, this is a preclinical model of MS in which the mice sort of develop inflammation-mediated demyelination through this autoimmune mechanism. So the pathology of the pathogenesis is similar to multiple sclerosis. And we have found that if we treated our mice, these mice, with the drugs that we have found, we're also able to promote repair. So whether it's complete, we see significant repair and recovery. but whether or not it's complete, we don't know. We are sort of still in the process of testing and characterizing these mice, and as well as doing more experiments to sort of figure out how much recovery is occurring.
It sounds, as I said, it sounds incredibly exciting. And I guess the Weaver Award is a 5-year grant to pursue this research. So I know we're going to be anxiously looking on and waiting for, hopefully what are going to be encouraging updates. But I know regardless of ultimate outcomes, we're bound to learn a lot about furthering our understanding of remyelination, demyelination. So I appreciate everything that you're working on, everything that you and your team are doing. I understand you even found time to participate in this year's Walk MS in Washington, D.C. Is that right?
Yes.
Well, we appreciate all the support on every level. And I appreciate you taking the time to talk with me today.
Well, thank you, John. I just want to say that, I've been very much involved in MS research now for probably the last 15 years. I'm grateful to the support from the MS Society, as well as from various foundations that supports multiple sclerosis research. And I think this has been instrumental in helping us pursue the research projects that we're working on. I'm always thinking about MS pathology when it comes to experimental design, because it's a fascinating, to me, it's the disease pathology is fascinating because of the sort of the complication. It's because it's so complicated in many ways. So it's a puzzle that we as scientists are interested in solving. And I would say that something that's pretty exciting about the work that we have done so far is that the molecules that we are looking at there are indeed drugs that target them, and some of them have already been approved, for other diseases. So it is very possible that we could translate some of our findings to patients, hopefully soon, at least to see if these drugs could potentially promote repair and recovery in multiple sclerosis. So I'm very hopeful and optimistic that we're going to get somewhere. And also the MS community, in terms of research, the researchers in the MS community are doing a lot of great work, and there's going to be a lot of interesting drugs in the next few years that's going to be potentially helpful to promoting repair in multiple sclerosis.
That's all just great news for all concerned. And I think I want to help my listeners understand a little bit when you can talk about a drug that's already been approved for use in maybe other diseases that you're finding has an impact on MS. What that means is we don't need to wait for another decade's worth of clinical trial work because we understand the safety already. That's been proven in previous clinical trials, which means people can actually get that treatment much faster than if it were something brand new. Do I have that right?
Yes, that's exactly right. I mean, there are already many drugs out there that might be used for other indications or other diseases. And then some of them may target similar pathways that are involved in multiple sclerosis pathology.
One of the things I really love about having conversations like this with people like you is that you can't help but come away feeling really hopeful about the future in terms of being able to unravel some of the riddle of MS and finding better treatments and one day being able to say we've got a cure. And this has certainly been a very, very hopeful conversation. So thank you again, Dr. Huang, for taking the time to take us into your lab and into your work. Thanks again.
Thank you so much, John. I appreciate, the conversation and discussing our research. Thank you.
Before we wrap up today's episode, I want to thank each of you who took the time to submit the story of your MS journey. After reading every one of them multiple times, I can see that I'll be reaching out to a few of you to share your stories in future episodes of Real Talk MS. But as for episode 100, my special guest is going to be Jody Michelle Cooley. When she was 10 years old, Jody's mom was diagnosed with MS, and as her mom's MS progressed, Jody transitioned from child to MS caregiver. Then at the age of 34, after giving birth to her own daughter, Jody received her own MS diagnosis. So in a few weeks, I'll be talking to Jody about her dual roles as an MS caregiver and as someone who's living with secondary progressive MS today. I'm John Strum. Thanks for listening, and we'll catch you next time.